Hello, everyone. Thanks for coming to my talk today, where we're going to be discussing the mechanistic basis of action of serotonergic psychedelics within the framework of the neuroplastic hypothesis of psychedelic action. We'll discuss some background and some previously existing data, and also data from my labs, really trying to work our way down from these bigger behavioral changes we know so well to probe changes at more of a micro level, at micro level neural changes. So to start, why am I interested in understanding the mechanistic basis of these drugs? And why should we all be interested in this? Well, in recent clinical trials, serotonergic psychedelics such as psilocybin, and I say serotonergic to indicate that these drugs are active at the serotonin 2A receptor, have shown efficacy as rapid and long-lasting treatments for disorders such as depression, anxiety, and even substance use disorders. Current research suggests that a single dose of psilocybin in patients with cancer and clinically significant depression and anxiety is over 4 times more effective than traditional antidepressants, with a very rapid onset and a large placebo-normalized antidepressant effect up to 5 weeks post-dosing. So I mean more effective in the way that they treat symptom severity and also the longevity of these drugs. There are also similar results for substance use disorders. In a study looking at psilocybin-assisted psychotherapy in the treatment for alcohol dependence, patients significantly increased in their abstaining behaviors following treatment. And these significant effects lasted for up to 36 weeks post-dosing session. So despite all of these amazing results, and these are by no means exhaustive, this is not an exhaustive list of all the results that we've been getting out of clinical trials. Despite this, I do want to point out a gap in our knowledge. The mechanism of action by which these behavioral changes are occurring is not clear to us. These trials were also psychotherapy linked. And here I have a picture of a clinical therapy room from UW-Madison's clinical trials. These studies are psychedelic assisted psychotherapy paradigms. And so there's this inextricable link between therapy and behavioral change in all of these studies. This points out a huge challenge with human clinical trials because we're unable to clearly isolate biological changes occurring from drug action itself from those psychological changes that might be affected by the psychotherapy itself. So in our experiments, we're examining the mechanism of action of classical psychedelics in an animal model. And this is going to allow us to isolate those changes having to do with drug effects as opposed to just psychotherapy. So to probe at the micro level, let's first understand those macro level behavioral changes that are occurring. So at the level of behavior, it's well documented that in cases of stress-induced psychiatric disorders, such as anxiety and depression, cognitive flexibility is decreased or impaired. Now, cognitive flexibility is traditionally defined as the ability to adjust or modify your cognitive content of your mental set and behavioral strategies in response to changes in the environment and environmental demands and stimuli. So it's really the ability to update your thinking and adjust your behavior according to whatever feedback you may be getting from the environment. And interestingly, in a study done by Das and colleagues, it was found that psilocybin-assisted psychotherapy actually increased cognitive flexibility. So here in this graph, we have 2 panels, panels A and B. And in panel A, we see depression scores on a depression inventory called the HAM-D. So this is telling us something about the clinical severity about an individual's symptoms. And on the x-axis, you'll see the baseline point, and that is baseline. That's before anyone had received the psychedelic treatment. And then 1 week and 4 weeks is going to denote 1 week or 4 week post-dosing. So on graph B, that's where I want us to look, is we are looking at the amount of perseverative errors made by these participants on a PCET task, the pen conditional exclusion task, which is a task of executive functioning. A perseverative error is an error that an individual makes when faced with a subsequent question or demand from the environment. So you have a task, you answer it and you get it wrong, you commit an error, and then you're given something else and you commit the same exact error. So this shows that there was a lack of adjusting and a lack of shifting your mental set, lack of changing your behavior. So we can see that as cognitive inflexibility. So here we can see that the rate of perseverative errors for most participants after dosing decreased. So we can take that as that cognitive flexibility actually increased. So why is this interesting and important to us? Cognitive flexibility can actually be thought of as a form of behavioral plasticity. And we're talking about trying to understand the mechanistic basis of psychedelic action. That's what we're trying to do here. So using this, we can really talk about a hypothesis in the field, which is the neuroplastic hypothesis of psychedelic action. So what's this hypothesis? The neuroplastic hypothesis of psychedelic action asserts that these compounds may be a catalyst for behavioral change through their action as psychoplastogens, or drugs that are able to induce neural plasticity. So neuroplasticity is the capacity of the brain to form new neuronal connections, both functional and structural, to grow, prune, and reorganize in a way that is optimally efficient. So there's actually evidence that psychedelics promote this type of plasticity in various ways. At the molecular and genetic level, single administration of serotonergic psychedelics have demonstrated the ability to induce transcription of various plasticity-related genes and immediate early genes, such as those that encode for BDNF, brain-derived neurotrophic factor, NMDA receptors and AMPA receptors, which you see there inserted into the membrane, that are very important for plasticity. And these changes in transcription occur globally, but also significantly in areas such as the medial prefrontal cortex and in the hippocampus. At the cellular level, serotonergic psychedelics have been evidenced to induce synaptogenesis within cortical areas. Synaptogenesis within cortical areas. So increasing synapse size and number, as well as insertion of more receptors into that synaptic membrane. These compounds are similarly evidenced to increase spine density, stability, and growth within cortical areas. So you'll see the number of spines grow on neurons. And further, there's also evidence for neurogenesis in the creation of new neurons occurring in cortical areas, such as the medial prefrontal cortex and the hippocampus. This has also been evidenced to be accompanied by lower action potential thresholds of cells in the ventral hippocampus. So this is going to indicate some level of enhanced functional plasticity, as well as that structural plasticity that we just talked about. So with that, now we see that psychedelics actually exert their psychoplastogenic effects at various levels. We've discussed evidence that shows that single administration of psychedelics, such as psilocybin, increased transcription of plasticity-associated genes. They increased dendritic spine density and increased synaptogenesis, as well as spinogenesis within cortical areas. And also, we see that there are increased connections right at the circuit level. I just want to point out that all of these data that we have are occurring in vitro in sort of cultures, in cell cultures. So taken together, these data do provide a good foundation for exploring plasticity as a crucial mechanism for change. However, there are still some significant gaps in the literature and some critical questions remain unanswered. So no study has looked to examine these electrophysiological changes in plasticity in vivo in behaving animals in both in cortical areas over a long period of time. Additionally, these changes in plasticity have not been examined in the context of pathology or in circuits that are relevant to the pathophysiology of psychiatric disorders. Finally, this vital connection between those macro level behavioral changes and these micro level neural changes has not been directly tested and sort of experimentally evidenced. So to understand these neural changes in the context of circuit level changes and working our way to a more micro level, we have to key into a specific circuit, right? So we're going to key into a circuit from the ventral hippocampus to the medial prefrontal cortex. And this is what it's going to look like here. So it's going to travel from the ventral portion of the hippocampus to the medial prefrontal cortex. And there are actually direct afferents from the ventral hippocampus to the medial prefrontal cortex. And this is what it looks like in some beautiful images that we have here. So it has been evidenced that there is actually weakened connectivity in this circuit in humans and in rodent models in various psychiatric conditions, or in the case of rodent models, stress-induced sort of phenotypes and acute threat responding. This circuit also exhibits changes in plasticity in response to pharmacological intervention, as we saw earlier. And this makes it a really great candidate to focus my experimentation. And further, as I just mentioned before, there are direct afferent connections from the VCA1 to the medial prefrontal cortex, along with some significant expression of those 5-HT2A receptors at which the serotonergic psychedelics are going to be active. So now we figured out what we're looking for, which is going to be plasticity. We figured out where we're looking for it. It's going to be in this VCA1 to MPFC circuit. So now we have to ask the question, we're poised to ask the question, what is the role of synaptic plasticity in the mechanism of action of serotonergic psychedelics in the hippocampal-prefrontal cortical pathway? To answer this question, I conducted electrophysiological experimentation in vivo in freely behaving mice and recorded electrophysiologically under drug and non-drug baseline states. So let me walk you guys through how I did that. So to start, these experiments were in vivo, and because they were in vivo, we have to conduct a stereotaxic surgery in which I chronically implant male and female C57BL6J mice with a bipolar stimulating electrode in the ventral CA1. So I want you guys to look at that bottom slice there on the right side. That's a bipolar stimulating electrode in the VCA1. And also I implanted 3 tungsten twisted wire recording electrodes bilaterally in the medial prefrontal cortex and also contralaterally in the VCA1, contralateral from that stimulating electrode. So now once this happened, we allowed the animals a week to sort of recover from their surgery and make sure everything was okay. And then we conducted experiments. In my experiments, I conducted data at multiple time points, starting with day 1 or the baseline day, in which I took various baseline recordings, including a stimulus response curve. So the stimulus response curve consisted of applying stimulation at varying amplitudes going from 0 microamps all the way up to 600 microamps. So again, applying stimulation to that ventral CA1 location. And after I did this, I recorded from those medial prefrontal sites as well as that contralateral site. After conducting those baseline recordings, I commenced with injections. So I had various groups. I had a saline group, psilocybin, which is a 5-HT2A psychedelic, 4-ACO-DMT, which is also a serotonergic psychedelic, which actually converges on psilocin, DOI, which is also a serotonergic psychedelic, and then 6-fluoro-DET, which is serotonergic. It's a serotonergic DMT analog, but it is not hallucinogenic. It does not have any hallucinogenic activity like the other drugs. So after these initial, after these drug recordings, I did more stimulus response curves at 24 hours and then 48 hours. And what did I find? So here I have group data across all of my animals. And on the x-axis, you'll see the drug, and on the y-axis, you'll see fractional change from baseline. So we see here that after psilocybin administration, the maximal evoked response, so the highest response that we were getting in those stimulus response curves, was about 20% higher than at baseline. And for 4-ACO-DMT, it was about slightly more than that. For DOI, it was around 10% higher. And interestingly enough, for 6-fluoro-DET, our non-hallucinogenic DMT analog, it was around 16% lower. So this is a very interesting result here, and we postulate that this can be due to the biased agonism happening at the 5-HT2A receptor. It's a G protein coupled receptor, and so ligands binding here, anything binding here, can have significantly different or biased downstream mechanisms based on the residues with which they're interacting on the receptor itself. That is something that we need to explore further. And also, I do want to point out that this is preliminary pilot data, and I am working on increasing the Ns for all of my groups, but nevertheless, still a very interesting result that we have here. So to sum up this electrophysiological result, we find that 24 hours after administration of serotonergic psychedelics, there's increased maximal evoked responding in the medial prefrontal cortex. So now we were able to sort of find a microcircuit level change having to do with synaptic plasticity in response to psychedelic administration. With these results, I hope to add to the field and contribute to the field in elucidating the mechanistic basis of action of these drugs, right? So I've added a small piece of data. Hopefully I would like to add more, but I would really like to drive the point home. I really want to make this a big point. It's so important that as a field, we work from going from this macro view, right? Macro view at the level of behavior. We see that there's changes in cognitive flexibility. We see that there's changes having to do with symptom severity across the board. Brain-wide changes, we've had some great results coming out of labs having to do with EEG and sort of functional MRI results, right? Global brain-wide changes. I want to work us towards more of a micro view at the level of circuit level changes, right? Circuit level changes and also cellular level changes. It's pretty evident to us by now that there is some incredible capacity for these drugs to induce change, right? But only by understanding how these drugs work at the most basic level will we be able to maximize their therapeutic potential and thus maximize their incredible potential for change. With that, I'd like to acknowledge members of both of my labs and everyone here at PsychedelX. This has been a really, really amazing process working with everyone and learning from you guys. So thank you so much for listening to my talk and attending here.